Okay, welcome all of you students. Today in this video, we are going to see the genetic engineering practicals. Okay, so we are going to see, or I'm going to explain the four practicals, that is molecular size determination of DNA in agarose gel electrophoresis. Then after that, we are going to see Western blotting, and then we are going to see Southern blotting, and then cDNA cloning, okay, or cDNA cloning by RT-PCR. That is RT-PCR here is called as reverse transcriptase PCR. So that we are going to see. Okay, so we are going to see in details about each practical. So I hope all of you are getting my voice. Okay, so let's start with the explanation here. So first practical that we are going to see is molecular size determination of DNA. So let's start with the practical, first practical, that is molecular size determination of DNA. Here in this practical, we are determining the size of DNA using a ladder. Okay, so in this kind of practical, the size in the size determination, the first procedure that is done is agarose gel electrophoresis. Okay, so the first process that is done here is agarose gel electrophoresis. So in this, what happens, you are having the samples. Okay, for example, I am having 3 DNA samples. Okay, suppose I am having 3 DNA samples, sample A, sample B, and sample C. Okay, sample A size is 10 kb, sample B size is 25 kb, and sample C size is 40 kb. Okay, so A equal to 10 kb, B equal to 25 kb, C equal to 40 kb. Okay, so these are the three sizes of DNA. So we actually don't know the size of this DNA, suppose. Okay, we have got three samples here, A, B, and C, and the size of this DNA we want to determine using the DNA molecular size determination practical in molecular biology or genetic engineering. Okay, so first of all, first procedure that we are going to do is agarose gel electrophoresis. Plus, there is one DNA sample that is present in the various types of kits, or that can be procured commercially. This sample is having this sample is called as DNA ladder. Okay, so what happens here in this DNA ladder? It is a DNA sample which is having the different bands of DNA. In this kind of sample, for example, there is a ladder. Okay, so particularly for this determination of this DNA, I know that the DNA obtained, that is ABC sample, is not a larger DNA. It is obtained from generally a bacteria or a virus. So I know the range of the DNA that is available in this kind of microorganisms. Okay, so I am going to take a 100 kb ladder here, 100 kilobase per ladder. This means that this DNA ladder that I am using, or DNA sample that I am using to determine the molecular weight, will have around 10 bands. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. There will be 10 bands. Okay. So the band which is when we put this ladder, this 100 kb ladder into the agarose gel electrophoresis, what is going to happen? This ladder is going to run. Okay. We are going to do the electrophoresis. Okay, so 100 kb ladder, so there will be maximum molecular weight. So the maximum weight of 90 kb. Yes, will have a molecular weight of 90 kb. And then the next band is 80 kb. Next band is 70 kb. Then 60 kb. Then 50 kb. kb means kilobase per. Then after that 40 kb. Then 30 kb. And then 20 kb. And then 10. Okay, so the last or the which is moved further from the agarose gel electrophoresis well will have a 10 kb ladder. Okay, so this kind of sequence of different bands you are going to get in the agarose gel electrophoresis when you are going to load a DNA ladder. Okay, so let's draw a diagram of agarose gel electrophoresis here on this side so that you can. Okay, suppose this is the agarose gel. Here let me draw a. Suppose this is the agarose gel. Here there are 4 wells here. Example. Okay, so the example which you are taking here is having 4 well. Okay, so this is the first well, second well, third well, and 4 well. Okay, so this is the agarose gel which is having a 4 well. Now we are going to add the DNA ladder in the first well. Okay, so as you know, the DNA ladder is of 100 kb that we are using here. You can choose the amount of DNA ladder or the size of the DNA ladder according to the DNA which molecular weight you have to determine. Okay, so I am taking here the 100 kb ladder. What is going to happen? This 100 kb ladder is loaded onto this well. Okay, so first lane will be of DNA ladder. In the second lane, you are going to add sample A. In the third lane, you are going to add sample B, and in the fourth lane you are going to add sample C. Okay, so what is going to happen here? Remember, when you do the agarose gel electrophoresis, the bands in the DNA ladder starts to move. Okay, so it will move like this. Okay, so these are the 10 bands that is obtained when you do the agarose gel electrophoresis of around agarose gel electrophoresis of this DNA ladder, that is 100 kb DNA ladder. Okay, so what is going to happen here? There are 10 bands that is obtained here. Okay, so now when you get the 10 different bands, the sample will also move along with that electrophoresis. Okay, so we know that the sample A here is of around 10 kb. So the sample A lane will form a band right at the ahead place. You can see here. I will draw the well for this with different color. This is the sample A. Okay, so the band which is formed here by the sample A will be right at the adjacent side to the 10 kb band of the DNA ladder. Okay, then the next band that is formed will be of band B in the third lane. Okay, third lane. In third lane we have loaded the sample P here. Okay, so sample P will form a band here right between the 20 kb and 30 kb band. Okay, so you can see here the first band here in the DNA ladder is 10 kb, then the second is 20 kb, then 30 kb, then 40, 50, 60, 70, 80, 90 and 100. Okay, so these are the different 10 bands. Okay, so accordingly the sample A, B and C will be eluted or will be moved in the agarose gel. You can see here the sample C is going to form a band right at 40. Okay, okay, so this is the agarose gel that we are going to get here. The three samples, that is A, B and C, are going to form its respective band or move according to the electric field that is applied uniformly to all the 4 wells. Okay, so what is happening here? The sample A will align itself with the first band of the DNA ladder, that is 10 kb. Then sample B is going to align it with 25 kb, that is in between 30 and 20 kb. So the band will be in between 30 and 20 kb, and the C part or the C sample is going to form a band adjacent or along the 40 kb. Okay, so just by seeing that we cannot determine the exact molecular weight here, so we have to use a formula called as relative flow formula. Okay, I'll tell you what is relative flow, which will help us in determining the molecular weight of the three samples. Okay, so what happens, there is a formula called as relative flow. Okay, so relative flow here means that band formed by different it is band, just a minute, it is distance traveled by band divided by distance traveled by different. Okay, so what is going to happen here, remember, when the agarose gel electrophoresis is done, there are two dyes in that. Okay, so the two dye is going to form a different here. Okay, so generally the two dyes that are used here is, first one is bromophenol blue, and second one is xylene cyanol. Okay, so the two dyes that are used here is, first is bromophenol blue and second is xylene cyanol. Okay, so you have to note the bromophenol blue is going to form a different like this. Bromophenol blue will move up till here and then we have to stop the electrophoresis. Okay, so when the bromophenol blue dye reaches at this position, we have to record the reading, or we have to measure the reading, that is from the well, from the start of the well till the ending point of the diaphragm. So the bromophenol blue will move at a certain distance after the electrophoresis. So we have to record the distance traveled by the bromophenol blue dye, that is dye front, from the well to the end. Okay, so we have to record this. Okay, then after that we are going to see the distance of each band of this well of this DNA ladder. So we are going to determine the distance of each of this band from the start of the well. Suppose, for example, suppose we are determining the relative flow for the first band, that is 10 kb band. Okay, so the suppose the diaphragm has traveled here 5 centimeter. Okay, so I think you have got this formula, that is relative flow is equal to distance traveled by band divided by distance traveled by diaphragm. Okay. Okay, so what are we going to get here? We are going to get the RF value. Okay, so first of all you have to determine the RF value for RF value for the band number first. Okay, so for example the diaphragm has moved 4, suppose diaphragm have moved 6 centimeter in the agarose gel. This diaphragm have moved 6 centimeter in the agarose gel. Then what is going to happen? The first band has moved, suppose 5 centimeter. Okay, so the relative flow will be 5 by 6. Okay, so you have to divide this value and you are going to get the particular relative flow of this. Okay, so likewise you have to determine the relative flow of each and every band in the experiment. Then after getting the values of each and every band, that is relative flow of each and every band of the DNA ladder, then you are going to plot this graph in a very specialized graph paper that is called semi-log graph paper. Okay, so the graph that is used here is called semi-log graph paper. Okay, so what is the this is not a normal graph paper. What happens here? Here on the x-axis, here, the readings are in normal scale. Okay, on the above axis, that is y-axis, on the y-axis the readings are on normal scale, and on the x-axis the readings are in logarithmic scale. Okay, so half of the scale here is logarithmic scale and half the scale here is normal scale. Therefore, such kind of graph is called semi-log graph paper. Okay, so what happens here? The scale here, logarithmic scale, will be used for molecular weight. The logarithmic scale here will be used for molecular weight. The normal scale will be here used for relative flow. Okay, so what happens when you plot this, all these values or relative flow of all the 10 bands, you are going to get a particular, shape like this. Okay, and then you have to plot the intercept for sample A and get its molecular weight. Sample A will be like this, sample B like this, and sample C will be like this. Okay, so you can determine the molecular weight of sample A, B, C like this. Okay, so in this way, this molecular size determination of DNA uses the semi-log graph paper, from which you can determine the exact molecular weight of different samples of DNA using the DNA ladder. Okay, so the DNA ladder that you are using here will be very specific. The DNA ladder should be according to the weight of the sample that you are using here. Okay, so whatever the sample's molecular weight that you are determining, the DNA ladder should be like that. Okay, so this was all about the molecular size determination of DNA by using DNA ladder and relative flow method. Then we are going to discuss the next one, that is southern blotting. Okay, so let's proceed with the southern blotting. Actually, next is western blotting, so let's proceed with the western blotting. Okay, so here western blotting means the determination of protein here. So detection of protein band from a proteome of a organism. Okay, so in this you have to determine the proteome of an organism. How you are going to determine the protein band, that is, or a protein, enzyme molecule or a protein molecule that is present in a particular organism or not. Okay, okay, so let's see now the next one. So what is done here in this western blotting? First of all, the starting of Western blotting is with SDS page. Okay, so you perform SDS page where you have to determine a particular protein in a proteome of an organism. Proteome means the total protein content or the total protein that is expressed in an organism at that particular time. Okay, so first of all, what you are going to do, the first process in the Western blotting is isolation of total protein content. Total protein content you are going to isolate of a particular organism, and then you have to denature it, okay, by boiling. Okay, so when you boil and add the sample loading dye, the protein is denatured as in SDS page the sample should be in primary structure. Okay, so now what is done here? Okay, so first of all, SDS page, let's see what is SDS page. SDS page means sodium dodecyl sulfate. Sodium dodecyl sulfate, polyacrylamide, polyacrylamide gel electrophoresis. Okay, so this in this electrophoresis there is presence of two monomers, that is acrylamide and bisacrylamide. So this acrylamide and bisacrylamide, upon the addition of two reagents such as APS, that is ammonium persulfate, and TMAID, polymerizes into a gel. Okay, so on the addition of this APS, that is ammonium persulfate, generally the concentration is 1% here of both of them, ammonium persulfate and TMAID. When you add this ammonium persulfate and TMAID into the gel or the mixture of acrylamide and bisacrylamide, it polymerizes and forms the SDS page gel. Okay, so there are two types of gel here. First is separating gel and second is stacking gel. Okay, so the stacking gel stacks all the proteins together. So when you see SDS page here, I'll show you the gel of SDS page. Suppose this is the gel of SDS page here. So there are two separate gels here. The upper gel here is called as stacking gel, and the lower gel here is called as separating gel. Okay, so what happens here? The proteins or the different wells of the proteins are made on the stacking gel, and this stacking gel runs the protein and it is getting separated on the stacking gel. Okay, so we are going to run the protein sample or the total isolated protein sample in two different wells. What will happen here due to this? The proteins will be separated. Okay, so after the protein are separated, the number of proteins are present in this total sample. So at a particular time, the different samples or the different proteins that are being present will be in large in number. So due to that, what is going to happen? We are going to detect one of the protein out of that. Okay, so for that particular for detecting that particular protein, we are going to use a probe. Okay, or we can detect, or we can do the detection here with without the probe also. There are two types of detection here. It can be called as screening also. There are two types of detection of screening. One is radio labeled, or second is non-radio labeled. Okay, so in radio labeled detection, what is done here? We are using a radio labeled probes, and then after that auto radiography is done. So the probe binds to a specific protein of that separated proteins on the SDS page gel. Now, when the probe has bind to the specific proteins in that SDS page gel, then you are going for autoradiography, and in that particular protein, in that particular gel, suppose this was the gel. Before detection, what are you going to do? Suppose this is the protein bands. So first of all, you have to go for electro transfer. Okay. So what are you going to do? You are going to cut this portion of the SDS page gel and stick it along with the nylon membrane. Okay. Nylon membrane or nitrocellulose membrane you can use before going for detection. Suppose I have cut this part of the SDS page gel onto which the proteins has been run. Okay, so there will be different number of bands of the protein onto this particular well. So I am going to cut this particular well and I am going to prepare a sandwich of the nylon membrane, and suppose this is the nylon membrane and this is the gel. So in SDS page, the proteins is having the negative charge. Okay, so nylon membrane is always placed onto this side and it is transferred in a electro transferring assembly. So in this electro transferring assembly, there will be a positive charge on this side and there will be a negative charge on this side. Okay, negative charge. Okay, so what is going to happen here? The proteins are negatively in charge. Negative charge proteins from this SDS page gel is going to transfer onto this nylon membrane. Okay, so this is called as electro transfer. This method is called as electro transfer. Okay, so due to this, what is going to happen? All the protein bands are going to transfer onto the nylon membrane. So once it gets transferred onto the nylon membrane, you are going to follow different procedures in order to fix that protein bands onto the nylon membrane. Okay, so after the blotting, the nylon membrane is subjected for baking at 70 degrees centigrade. Okay, so after the baking, what will happen here? The protein bands are immobilized. To fix it, fixing is done under UV rays. Okay, so when you subject this under UV rays, the protein bands will be fixed onto the nylon membrane, and then after that you can go for detection. Okay, so detection is done either two ways. I already told you. First is radio labeled detection and second is non-radio labeled detection. In the radio labeled detection, what is done? Radio labeled probe is used and then the detection is done, and in non-radio labeled detection you are going to use different intermediate compounds such as biotin or different antibodies that are very specific to certain proteins. Okay, so in case of SDS page here in Western blotting, we are going to use different antibodies that are binding to the probe. Okay, for example, suppose this is the nylon membrane, which is having the different number of protein bands. Okay, so these are the different protein bands that are present onto the nylon membrane, and you have to detect a particular band here. So for what is done, a probe molecule is used. This probe molecule is having a antibody with it. Okay, so this antibody here is called as primary antibody. Okay, so now when the probe molecule is attached onto this gel, it is this membrane is washed along with the solution of this primary antibody along with the probe. Probe plus primary antibody solution is added. The probe is going to bind very specifically at a particular protein site here. Okay, so for example, the probe is binding to this band here. Okay, so this is having antibody like this on both sides. Okay, so after that you are going to add, or you are going to do the treatment of secondary antibody. Okay, so after that you are going to add secondary antibody here. Okay, the secondary antibody is going to bind at this position like this. Okay, and then the secondary antibody is labeled with a particular enzyme. Okay, the secondary antibody is labeled with a particular enzyme. The enzyme that is used here is generally HRP, that is horseradish peroxidase, or it can be alkaline phosphatase. Okay, it can be AKP, that is alkaline phosphatase. Now these two enzymes are used here. Suppose we are using the HRP enzyme here, that is horseradish peroxidase, that is HRP plus secondary antibody. Okay, so if that core probe molecule has binded to a particular protein here, the primary antibody will go and bind at this position, and we are going to do the subsequent wash. Then after that, the secondary antibody, which is labeled with the enzyme, is going to bind at this position, just like we do in ELISA. And then the secondary antibody is added with the substrate molecule. The substrate molecule here for HRP is TMBH2O2. Okay, so when the enzyme acts on the substrate molecule, it is degraded and forms TMBO plus O. So this TMBO is blue in color, which gives a blue color to this area. So this whole area becomes blue in color. Okay, so thereby we can say that here the band which is present here at this position is the band which we are going to detect. Okay, so we have at the starting only I told you that we have run the sample in two different copies in two different well. We have taken one well for detection, and now we are going to compare this nylon membrane with the well that is present, extra well that we have took at the starting. Okay, so from that what we can do, we can compare. Suppose this was the gel piece, this is the gel piece here at the starting. Okay, and these are the different bands that are present, protein bands. So out of that we can tell that this is the band, that is fourth number band that is present in this protein sample. So thereby you can confirm whether a particular gene that has been cloned in the organism has expressed in that organism or not. Okay, so if that particular protein is expressing, then only it is going to form a band here, and then we can detect that. Okay, so this is how the whole process of Western this is how the whole process of Western blotting takes place. I think you are understanding it. Is there any question? Okay, so let's move further. The next type of practical that generally is done in genetic engineering is the Western blotting. Southern blotting. In Western blotting, we detect protein. In Southern blotting, we detect a particular DNA that is present in an organism or not. So let's see the next one, that is Southern blotting. Okay, so in Southern blotting what happens, the whole process is same. First of all, you are going to I will enlist the step. The first step is isolation of total genomic DNA. Okay, suppose in this DNA, suppose there is any bacteria, so in that bacteria we have added a gene into that. Okay, so we have to check whether our our interested gene has entered into that bacteria or not. Okay, so we have to check that. Okay, so in order to check that, what are we going to do? We are going to subject it for the total genomic DNA from such kind of bacteria. Okay, so in this restriction digestion, the DNA is made into different fragments. So after that, what is done here? So for example, I will tell you here. Suppose after doing the restriction digestion, suppose this is the gel. Okay, this is the gel, and the DNA sample is run in two different wells. Okay, so there may be more wells, but for just for the sake of convenience, I am showing you here two different wells. Okay, so like this, the whole DNA sample is loaded onto the agarose gel and the electrophoresis is done. Okay, and you are going to make two copies out of that. Okay, so you have to cut the gel in between like this and use this copy for southern blotting detection and this for comparing. Okay, so we have to take this copy now. Okay, so now the next step in southern blotting is electro transfer. So what is done in this electro transfer? There is an electro transferring assembly like this. Okay, the electro transfer assembly is having a positive electrode and here there will be negative electrode here on this side. Okay, so in this here what is going to happen now? So you have to first place the nylon membrane here on this side, suppose, and here there will be here there will be suppose this is the gel, this is nylon. So nylon membrane is having positive charge and DNA is negative in charge. So DNA will bind to this membrane. So there is an electric field that is supplied here. Okay. So now what is going to happen? That all the DNA bands will be blotting onto this side and DNA bands will be blotted onto the nylon membrane. Okay. So this generally takes place. This generally takes place for about it depends on the DNA also. This generally takes place for about 3 hours. So depending on the size of the DNA. Okay. So what happens? Let's see what happens here. So after the electroblotting, then we have to go for detection. Similar to the Western blotting, here also there are two types of detection here. The first is radio labeled and second is non-radio labeled. Suppose this is the nylon membrane that we have taken and these are the different bands that has been blotted onto this membrane and we have to detect the particular gene of interest that we have added in our organism. So naturally this DNA will have that particular band. So this southern blotting is used to confirm whether our particular DNA has entered or is present in that organism or not. So we have to check that here. Okay. So how are you going to check? You are going to do either a radio labeled detection or radio detection. radio isotope detection or non-radio labeling, radio label detection. Okay, so there are probes used here for the detection purpose. Okay, so what are, okay, so for example, the gene of interest, if it is present there, then this probe molecule should go and bind at this position. Okay, so in case of radio label detection, what you are going to do, you are going to radio label the probe and then you are going to detect, put that probe into this. Okay, so the probe will go and bind to the particular place. For example, suppose this particular band into that nylon membrane, and you can say whether your particular fragment is there or in that DNA sample or not. Okay, so if you are doing the non-radio label, then you can go for various types of detection, such as deoxygenin detection, then biotin labeling probe. Okay, so I am going to explain you here the biotin labeling probe here. For example, suppose these are the different bands here, and you are using the probe molecule here. Suppose this is the probe molecule here. Okay, and it is being labeled with biotin. Okay, so this probe added into this membrane in a plastic Petri plate, as DNA binds to the glass, so therefore here plastic Petri plates is taken, and now suppose this probe has bind at this position. Okay, suppose this probe molecule has bind at this position. Okay, for example, you can see here I am showing, so this is the probe molecule attached at this position. Okay, so then what is going to happen? What is going to happen here? Then we are going to do subsequent washes. Okay, so the washes here done with different buffers. So why these different buffers are used? To exclude extra probe molecule here, you have to use different buffers washes. The buffer can be here A, B, C, D. So all the function of this buffers is to wash the particular thing. Okay, so we are missing one thing here. So first of all, before labeling here, this membrane is baked at 70°C for 30 minutes. Okay, so this baking ensures the immobilization of the DNA bands, and then it is expressed to UV after that for 20 minutes. So the UV will form a cross linking with the DNA bands and the nylon membrane. So the nylon membrane will have all the DNA bands that is immobilized now. So after that, this probe molecule is added in radio label detection also and in non-radio label detection also. So now we were at the point where we have added the probe here into this membrane, and now what is going to happen? We are going to put the probe here, and the probe has binded at this position to this band. Okay, we have added the biotin labeled probe here. So this green colored marker shows here the biotin. Okay, so now you know naturally the biotin is having the affinity towards the avidin or streptavidin. Okay, so we are going to add a component such as called as streptavidin. Okay, so suppose I am showing here the streptavidin in a triangular form, and it is present along with the enzyme that is HRP. Okay, so when you add this particular streptavidin along with the enzyme HRP, it adds or it joins to this particular band or this particular probe. Okay, so you can see here that probe is going to bind at this position. Okay, so after that the HRP has binded at this position. So what are you going to do? You are going to add the substrate solution. Okay, so the same substrate solution we are going to use here, that is TMBH2O2. So this TMBH2, when acted upon the HRP enzyme, it breaks into TMBO and O. So this TMBO is blue in color. So here the blue color will be formed, and this blue color band can be detected. And after that, you can use this particular well from the side to compare. Okay, suppose you are comparing this band. So the third number band is a particular band of interest, and you can cut this portion and purify this particular band using silica purification method. Okay, so this was the everything about the southern blotting. I hope you are understanding this. Okay, so let's see the last experiment here, that is RT-PCR. RT-PCR means reverse transcriptase PCR. Okay, so what is done in this PCR? You have to detect a particular viral protein or viral sequence. So then you or you have to amplify that particular viral sequence. Then you have to go for RT-PCR. Okay. So first of all, what is done? That particular viral DNA, for example, this is the viral RNA here. So this viral RNA will have a poly A tail along with it. Okay. So first of all, this viral RNA is converted into cDNA using the enzyme reverse transcriptase. Okay, so this poly A tail which is present at the end of mRNA is used here. So in this particular PCR, first step is the formation of cDNA. So the first reaction which you are putting here is the formation of cDNA reaction. So in this reaction, what are you going to do? You are going to add a poly DT primer. So this will be a poly DT primer that you are going to use here, and you are going to add, suppose this is the 5' to 3', and this is 5' to 3' on this side. So the reverse transcriptase enzyme is going to form a, it is going to form a cDNA here. Okay, so this now cDNA forms here in the blue color, you can show here. So in the next step, you are going to degrade the DNA. Okay, so now the cDNA is the cDNA that is formed here. We have to the cDNA that is formed here is again converted into, suppose this is the cDNA that is formed here, having the TTT on this side, is again converted into regular DNA. Now this regular DNA is used for amplification in PCR. Okay, so there are two steps in PCR, RT-PCR. The first step is the formation of cDNA, that is complementary DNA from the viral DNA using the reverse transcriptase enzyme, and then formation of double-stranded normal DNA, that is here, that is used for amplification purpose. Okay, so now after that you have to go for all PCR reaction. So you have to add along with this all the PCR components such as tag DNA polymerase plus Mg2 positive ions plus DNTPs plus buffer. Okay, so all these things you are going to add that are required for PCR, and then you are going to amplify this particular cDNA in PCR. Okay, so after the amplification, you are going to go for Agarose Gel Electrophoresis. Okay, so generally the cDNA reaction, the formation of cDNA reaction is about 40 microliter in case of RT-PCR, and from that you have to take 5 microliter of the sample, and you have to add 35 microliter of PCR mix, which is having all the different components of PCR, and then make it 40 microliter for the amplification purpose. Okay, so once you get the amplification of the cDNA, you have to go for Agarose Gel Electrophoresis, and you can detect or you can quantify that particular cDNA amplification onto the agarose gel. Okay, so for this, generally this technique, that is RT-PCR, is used in detection of different viral DNA that is present in a mammalian DNA sample or a eukaryotic cell DNA sample. Okay, so generally you can you know that this technique is widely used for the detection of the coronavirus RNA into the human cells. So the test that is done here is RT-PCR test. Okay. So one particular thing here that I want to tell you that is COT value, that is concentration value here. See, remember the concentration of the virus is higher. So suppose the concentration of virus is higher. So in that RT-PCR, the value is you can detect the virus is more easily in these few cycles only. And if the viral sample is present in a very low amount into the human sample or the eukaryotic cell, then the virus is detected at a very later stage. Okay, so thereby you can detect the viral DNA using RT-PCR. Okay, so I hope you are getting all the details of the practicals of this genetic engineering that we have discussed today. If there is any doubt, do comment your questions. Thank you so much for joining, all of you. We are going to end this session now. Thank you for joining.